In the last lecture, I described for you Mendel's famous first experiment, the so-called monohybrid cross, which led him to deduce many of the fundamental principles of genetics. Let me remind you at the start of this lecture of Mendel's main conclusions. First, he deduced that traits must be coded for by discrete heritable particles, things we call genes today, and that these heritable particles were transmitted as independent entities across generations. Second, Mendel deduced that there must be different versions of each of these kinds of heritable particles, in other words, what we call different alleles for each gene, and he concluded that every individual has two such heritable particles, two alleles for the gene within them. Third, Mendel realized that alleles can interact with each other. Specifically, he identified the fact that one allele might be dominant over the other allele. And finally, Mendel figured out that the two alleles a parent has are separated into different cells when gametes are formed, an observation that we refer to as Mendel's law of segregation. Now, in today's lecture, we're going to build on our knowledge of Mendelian genetics, specifically in order to look at how sexual reproduction actually generates genetic variation in offspring. The question we'll begin with is this. What happens if we cross parents differing in two phenotypic characters, not just one? Last time, remember, we asked what would happen if we crossed parents that differed in just one character. Flower color was the example we used. Now imagine that we cross parents that have two different characters. For example, flower color and seed color. I mentioned last time that there are a number of dichotomous traits in pea plants that Mendel found, and seed color is one of the many. There are two seed colors found in pea plants. Seeds can either be yellow or green. Yellow or green, it's a dichotomous trait, nothing in between. Like flower color, this difference depends on just one gene that has two alleles. The yellow allele is the dominant allele, and the green allele is the recessive allele. Now we're going to use the same letter convention, the tongue-tying letter convention we used last time, to label alleles. And in this case, we'll call the dominant yellow seed color allele big Y or capital Y, and we'll call the recessive green allele lowercase or little y. Because we're looking at the transmission of two traits at the same time, we're going to call the cross we're going to talk about a dihybrid cross, hybridizing for two traits. Now, let's begin by supposing that we cross a female pea plant that has purple flowers and yellow seeds and is from a true breeding line. Remember, that means we can assume that this individual is homozygous for the alleles for those traits, in that case, the dominant alleles for both traits. And we're going to start with a male plant that has white flowers and green seeds, and thus is homozygous for both recessive traits. Okay, it would have a little p, and a little y allele. Now, what we're going to do is ask what happens if we cross those two individuals, just as we did in the monohybrid cross we did last time, except now looking at the fate of these two traits as offspring are produced. Now, given that both of these characters are dichotomous traits, and given that I've already told you that they have dominance with, in this case, the purple flower color and the yellow seed color being the dominant traits, we might be able to guess what would happen in the F1 generation. You would expect that all of the F1 generation, that's the offspring of those parents, are going to have purple flowers and yellow seeds. In other words, all of those F1 individuals will express the dominant phenotype for both characters. This is because we know from what we talked about last time that they all have to be heterozygous for both characters. We know that they received the gene or the allele for purple flower color and yellow seed color from one parent, the mother in this example, and they received the white flower color allele and green seed color allele from their father. So all of these F1 individuals, just as we saw in the monohybrid cross, are going to be heterozygous. In this case, heterozygous for both of the genes that we're interested in. And because we have a clear dominance of purple flower color and yellow seed color, we know that all of these heterozygous F1 individuals are going to be purple flowered, yellow seed colored phenotypes. Okay, so that seems to make sense based on what we know. Now, the interesting question is what happens when we cross these F1 individuals? Now, the question we're really asking here, what we're getting at, is when these individuals, the F1 individuals, make gametes, do the alleles for the two different genes we're talking about stay together, the ones that are inherited from the same parent, or do they move independently of each other? For example, we know that each F1 individual has one big P allele that it inherited from one parent and one big Y allele that it inherited from one parent, the same parent, the mother in our example, and each has a little p allele and a little y allele that it inherited from its other parent, the father in this example. When these F1 individuals make gametes, do the two maternally derived alleles, big P, big Y, always go together in a gamete, while the paternally derived alleles, little p, little y, go together into another gamete? Or alternatively, can these individuals produce gametes that mix it up, that might put one maternal allele in a gamete with a paternal allele, and so forth? Let me clarify this question by elaborating in a little more detail on these two possibilities. Now, the first possibility suggests that the alleles inherited from one parent will always stay together. That is, we say that they remain linked, in which case an F1 individual could only produce two types of gametes. They could produce gametes having both dominant alleles, that is, a big P, big Y gamete, and gametes having both recessive alleles, that is, a little p, little y gamete. Now, if this were the case, if the F1 individuals could only produce two kinds of gametes, then what we would expect in the F2 generation when we cross them would be pretty clear. We would have a four-celled Punnett square, as we did last time, that would yield only the same two phenotypes that we saw in the parental generation. Furthermore, we would have only purple-flowered, yellow seed individuals, which could be homozygous for that dominant trait or heterozygous for both traits, or we could have individuals that were homozygous recessive for both traits, and those would have white flowers and green seed color. Furthermore, just as we saw in our monohybrid cross, we'd expect the ratios of these phenotypes to be 3:1, with 3 times as many purple flower, yellow-seeded individuals as there are white-flowered, green-seeded individuals in the F2 generation. In other words, if the alleles for flower color and seed color, which are inherited from the same parent, are linked, they always travel together. And the results of the monohybrid cross in the F2 generation would look the same as if there were only one gene. Imagine a gene that was somehow responsible for producing or affecting flower color and seed color. It would be the same thing, as far as we could tell in our F2 generation from this dihybrid cross, if they're linked. Now, the alternative possibility for how an F1 individual might make gametes is that the alleles for different genes inherited from the same parent move independently when that parent produces its own gametes. In other words, we say that the genes would be unlinked. In this case, there are actually 4 kinds of gametes that could be produced by each of the F1 individuals, not just 2. Of course, we would expect to have some gametes that have a combination of the big P, big Y allele, and we'd expect some gametes that have the little p, little y allele. This is what we saw before. But we would also expect equal numbers of gametes having a combination of big P, little y, or little p, big Y. All 4 of these kinds of gametes would be equally likely to be produced. Now, we can use, again, the convention of a Punnett square that I introduced last time to figure out all the possible ways that the gametes produced by these F1 individuals, if they're making these 4 kinds of gametes, could be combined. It would be a much more complicated Punnett square. Well, not a lot more complicated, but a little more complicated. We'd need to have 4 rows and 4 columns, and we would have 16 cells. And we could do the same thing we did last time. We could figure out what all of the possible combinations of these alleles are when diploid zygotes are formed. In other words, we would have 16 cells that represented all of the combinations of genotypes produced when these alleles are combined. Now, those 16 cells actually would only yield 9 distinct genotypes. And that's because some combinations of alleles would actually yield the same diploid genotypes depending on how they were combined. And we don't need to go through the details here, other than to realize that there would be 9 unique genotypes in the F2 generation. And to remind ourselves that not all of these genotypes would have unique phenotypes. Remember, different kinds of genotypes can produce the same phenotype, especially when there is a clear dominance of one allele over the other. Again, we don't need to work out the details. I'll just tell you that if we looked at the 9 possible genotypes that were produced in the F2 generation, they would correspond to 4 possible phenotypes. We would have individuals with purple flowers and yellow seeds, both the dominant traits, which we saw in the parental generation. We would have individuals with white flowers and green seeds, both the recessive traits, which we again saw in the parental generation. We would also see, though, combined traits. We would see some individuals with purple flowers and green seeds, we hadn't seen before, and other individuals with white flowers and yellow seeds. Again, a combination that we hadn't seen before. Furthermore, if we worked out the details, we could predict that the phenotypes that we would see in the F2 of this dihybrid cross should occur in the ratio of 9 to 3 to 3 to 1. Now, working out the combinations and the probabilities isn't really the important point here. The key point is to realize that 2 of these phenotypes are completely new, in the sense that they never occurred in the parental generation. Specifically, the purple flower color, green seed color phenotype, that combination, and the white flower color, yellow seed phenotype, the other combination, represent combinations of traits that we had never seen before. We call these new combination of traits recombinant phenotypes. Well, now the question is, what did Mendel see when he did the dihybrid cross? Well, you might guess the reason I've been going on so much about this alternative possibility is that's exactly what Mendel observed. He found 4 phenotypic combinations in the F2 generation of his dihybrid cross, including both of the recombinant phenotypes as well as the parental phenotypes, occurring in the predicted ratio of 9 to 3 to 3 to 1. Now, using the same logic that he used when he was considering the results of his monohybrid cross, Mendel realized that the only possible explanation for these results was that the heritable factors, the alleles, responsible for these 2 different phenotypic traits completely independently assorted when these F1 generation individuals made gametes. This result led Mendel to formulate a second principle of inheritance, which we now call Mendel's law of independent assortment. And Mendel's law of independent assortment simply says that when we have multiple characters, they may completely move in the formation of gametes and combine in different ways. The significance of this, of course, is that it results in the appearance of phenotypes in later generations that we hadn't seen earlier in the parental generation. Let's look at this in a little more detail by going back to what we learned about meiosis. Because it turns out that what Mendel inferred from his crosses, we now could predict by looking at the movements of chromosomes in meiosis, something Mendel knew nothing about. Remember what happened during that first meiotic division we talked about a couple of lectures ago. Homologous pairs of chromosomes, those are the copies of the same chromosome type, the homologs that you inherit separately from your 2 parents. Those homologous pairs line up at the middle of the cell during the first metaphase, and the 2 homologs are separated into daughter cells when that first meiotic division is completed. Remember also that when this happens, the maternally and paternally derived versions of these homologous chromosomes, in other words, the homologs you get from one parent or the other, sort out completely independently. The way that one homolog arranges at that first metaphase has nothing to do with the way that any of the other homologous pairs are arranged at that first metaphase. In this way, the assortment of homologs is completely independent of each other across different chromosome types. Now imagine that the two genes responsible for, say, flower color and seed color occurred on two different chromosomes. We would predict, just as Mendel observed, that the alleles for these genes would assort completely independently when gametes are formed. So in other words, the independent assortment that Mendel saw when he looked at his dihybrid cross results actually matches exactly what we would predict if the genes he was looking at for these two traits occurred on different chromosomes. And the independent assortment that Mendel saw was caused by the random or independent assortment of different homologs during the first meiotic division. Now, this conclusion brings me back to the point I've made earlier about how sexual reproduction generates genetic variation. We began this dihybrid cross with just two phenotypes. Individuals in the parental generation either had purple flowers and yellow seeds or white flowers and green seeds. But we saw in the F2 generation the emergence of two entirely novel or recombinant phenotypes: purple flower green-seeded individuals and white flower yellow-seeded individuals. These phenotypic combinations had never been seen in the parental lines, and thus they represent the generation of a kind of variation in subsequent generations. Now, the amount of variation that could be generated by homologs separating independently in meiosis is actually enormous. In the example we just talked about in the dihybrid cross, we started with two genotypes in the parental generation in those true breeding lines and two phenotypes. And we ended up, by the time we got to the F2 generation, with 9 different genotypes and 4 phenotypes. Actually, the numbers of combinations of alleles on different chromosomes, if they occur on different chromosomes, can be actually staggering, depending on the number of chromosomes. So for example, humans, as I told you, have 23 homologous pairs of chromosomes. The number of different ways that the homologs for these 23 chromosomes could sort themselves out during gamete formation is actually 2 to the 23rd power. In other words, there's a coin flip done independently 23 times. If you do that and combine those probabilities, you realize that you can actually make over 8.4 million different combinations of the 23 chromosomes you have. About 8.4 million different kinds of gametes given the same set of chromosomes that you're starting out with. Now, the point I want to make here is just to illustrate that there's an enormous amount of genetic variation introduced simply by the independent assortment of chromosomes during meiosis. And this is what Mendel uncovered when he did his dihybrid cross and formulated his law of independent assortment. Now, I want to step back for a second and contrast this variation that we see coming from independent assortment as gametes are formed and combined with a kind of genetic variation that we've already talked about earlier in the course, namely mutation. Remember, mutations in the DNA occur when an error made in DNA replication goes uncorrected, or some damage that happens to the DNA goes unrepaired. Mutations essentially generate new alleles, using the terminology that we've introduced. Usually when a mutation occurs in a stretch of DNA, the result is a dysfunctional allele that no longer works anymore. But sometimes a mutation might result in a functional allele that works maybe as well, maybe even a little bit better than the original allele. Essentially, when mutation happens, we've added another allele, whether it's functional or dysfunctional, to the genetic mix. We might start out with some number of alleles, and when we have a mutation, if it remains in the population, we now have one more kind of allele. By contrast, what independent assortment does is not create new alleles, but instead create new combinations of alleles for different genes. Both mutation and independent assortment can yield new phenotypes in subsequent generations, but the way mutation does this is by fundamentally altering alleles, essentially creating new alleles, while independent assortment does this by shuffling alleles for different genes within individuals relative to each other. Okay, now I want to make one more point about how genetic variation is introduced when cells undergo meiosis. This is actually not a point that Mendel realized. And in fact, it's not even clear that he could have figured this out without knowing about meiosis, because the way that this kind of genetic variation is introduced really is specifically explained by a particular fact of how meiosis works. Now, let me set this up by sort of reminding you what we might conclude based on what we've said so far about the dihybrid cross. We understand now that if genes are on different chromosomes, they'll assort independently, and that independent assortment makes new combinations of alleles and gametes, and therefore new genotypes and potentially new phenotypes. But what if two genes are on the same chromosome? Now, the first thing we might expect is that if two genes are on the same chromosome, they should be linked. They should always move together, right? Because when those chromosomes separate in meiosis, the whole chromosome goes one place or the other. Or does it? Remember back to our discussion of meiosis. Remember that in the first prophase, the prophase of the first meiotic division, I told you that those homologous pairs of chromosomes associate very closely. So closely, in fact, that they will exchange parts of themselves, essentially exchanging chunks of each chromatid from one homolog to the other. If that happens, if an exchange, if a crossing over event, as we called it, occurs between two genes on the same chromosome, the result will effectively be the same as if those genes occurred on different chromosomes. They can assort independently. Now, when Mendel's work was expanded on, well after he lived in the early 20th century, geneticists largely made the assumption that if genes were on different chromosomes, they would be unlinked and they would assort independently. But if genes were on the same chromosome, they would be linked, and the traits for those genes would always move together as offspring were created. Actually, that presented kind of an interesting problem for geneticists because, as I said, it's hard then to tell whether the two traits you're interested in are somehow affected by the same single gene or two different genes, right? If genes are linked, if they always move together, how do you know physically that there are two stretches of DNA that independently somehow cause these traits to occur as opposed to just one gene that might affect both traits, which is possible. You could imagine a gene affects the expression of color both in flowers and in seeds. Whether the seed color is yellow or the flower color is purple isn't the effect of that gene. It's just whether it's one thing or another. This is possible. Well, relatively soon, as geneticists tried to work this kind of problem out, they came across some results from dihybrid crosses that didn't fit quite the results predicted by Mendel's work. Specifically, what they found was sometimes a cross would produce just a few recombinant individuals, but not in the expected ratio of 9 to 3 to 3 to 1, as you would expect if those chromosomes were on, or if those genes were on different chromosomes. For example, let me try to make this clear by giving you some specific numbers. If you had two genes on different chromosomes and you did a dihybrid cross, such as we did with flower color and seed color, and you sampled some individuals in the F2 generation to make it work out evenly, let's say you looked at about 160 individuals, you'd expect a 9 to 3 to 3 to 1 ratio. So what you would expect is that about 90 individuals would have the dominant trait for both of those characters. About 10 individuals would have the recessive trait for both of those characters, and about 30 individuals each would have the recombinant phenotypes, the two different recombinant phenotypes. What was found instead, however, sometimes, is an intermediate result, for example, where you would have actually mostly the two parental phenotypes. You might say have 110 of the dominant phenotype for both traits, and maybe about 30 or so of the recessive phenotype for both traits, which is approximately a 3-to-1 ratio, which is approximately what you would expect to find in a monohybrid cross or if the genes were linked. But in that same cross, you might find, say, 5 or so individuals having one of the recombinant phenotypes and 5 or so individuals having the other of the recombinant phenotypes. What's going on here? Well, it turns out that what's going on comes from understanding crossing over during meiosis. When homologous pairs exchange genetic information, they will create these recombinant gametes that produce recombinant phenotypes in an F2 generation. But because crossing over is a relatively rare event, it doesn't occur every time. And in fact, it only occurs rarely between any two genes. So if you do a dihybrid cross with genes that are on the same chromosome, you usually find that most of the individuals are the parental phenotypes, as you would expect for linked genes, but you also occasionally find just a few individuals that are the recombinant individuals, or that are the recombinant phenotypes. So the point I want to make is that even if genes are on the same chromosome, the mechanisms of meiosis make it possible that those traits can be shuffled as gametes are produced, adding even more genetic variation in sexual reproduction. Well, Mendel's crosses, which he did back in the 1850s, really established what we now consider to be the modern field of genetics, because he really worked out, without knowing anything about chromosomes or genes, he really worked out what must be going on. Interestingly, when Mendel lived, his work was not appreciated. In fact, Mendel only published one paper. He published a paper describing his results for all of these crosses in 1866. That paper was distributed to most of the major libraries in Europe at the time, and Mendel himself sent the paper to about 40 or so distinguished scientists. But it was largely ignored. There are a couple of reasons that Mendel's work might have been ignored. The first was the fact that Mendel was making his inferences about heritable particles without knowing anything about the physical basis of what those heritable particles could be. Chromosomes hadn't been identified. Nobody had a clue what those heritable particles could be. And at a time in biology when most work was very observational, people found it very difficult to get their minds around this very hypothetical construct of heritable particles that Mendel proposed. Also, Mendel's work, as we've seen, relied heavily on the mathematics of probability theory. Mendel was well trained in mathematics, and he used this math to make the inferences from his crosses to give him his insights into how genes and chromosomes must work. But scientists in the mid-19th century were relatively unmathematical, especially biologists. And it's probably the case that most biologists at the time who were interested in problems of inheritance simply couldn't understand Mendel's math. The result was that Mendel's work actually became forgotten. It was virtually unknown, maybe entirely unknown, for 40 years. It had been completely forgotten until sometime later in the early 20th century, when geneticists again began to see the patterns that Mendel originally described and relate those patterns to what then had become known about the movements of chromosomes in mitosis and meiosis. That led to a revolution in genetics, and it's that topic that we'll address in the next lecture.